So in this section of the course, we're going to talk about chromosomal inheritance. And chromosomal inheritance is basically how do we inherit these massive chromosomal diseases that are caused by large-scale mutations where you have a whole chromosome missing or too many chromosomes. In this section, we'll also talk a little bit about how sex is determined in both humans and some other organisms as well. And we're going to talk about these things called karyotypes, which look at a layout of all the chromosomes that exist in an individual, and we can get a lot of good information from that. So first, let's review chromosomes. So we talked about this previously, that DNA is not just sitting inside of the nucleus. DNA is wrapped around lots of different proteins called histones, and usually, even though it's wrapped around those proteins, it's still kind of a jumbled up mess, and it's called chromatin most of the time. And it's only during mitosis or meiosis that the chromosome, or that the DNA actually condenses into chromosomes. And it's just this kind of tightly packed package of genetic material that can be easily pulled apart and distributed to the new cells that we are making. Remember, we have 23 chromosomes from mom, 23 from dad, and each one of those has a homologous partner for a total of 46 chromosomes in our cells. If we were to take every single pair and pull them out and actually lay them out, we call that a karyotype. So if you look here, we have chromosome pairs 1 through 22. And then we have one pair of sex chromosomes, which is at the end. In this case, we have 2 X chromosomes, as you can see over in this corner right here. These are replicated. Each homologous chromosome has a sister chromatid with it. You can kind of see it if you look. So here we have, here's one of our largest pairs, chromosome pair 1, and each of these has a partner with it that is its identical copy. So we know it's been replicated. So these karyotypes are actually very useful. We can use them to diagnose specific diseases, specifically even in utero before a child is born, if we think that they are at risk of having a particular chromosomal abnormality. And we're going to talk about those different diseases in a little bit. First, we're going to talk about how sex is determined in humans. So in sex determination in humans is done through an XY chromosome system. There are 23 pairs of chromosomes total. 22 of those pairs are called autosomes, which are normal chromosomes that both males and females have. There are 2 sex chromosomes, the X chromosome and the Y chromosome. If you are female, you have 2 X chromosomes. If you are male, you have an X and a Y. So sex is actually determined by the father because the father has an X or a Y to give, as you can see over here in this bottom corner, and the mother has only X's to give. So it's the father who determines whether or not you're going to get a boy and be XY or a girl and be XX. So we do this just like we would do in normal Punnett squares. You can see if the father can give an X or a Y sperm, the mother can give an X or an X egg, and as a result you get a 50-50 sex ratio, which is shown in the population, right? About half females and half males, barring any other type of selective things. Like, for example, in China, when they had the one-child policy, they would actually selectively abort children that were female. And now they have a huge issue of having way too many males and not enough females for them to kind of pair with. So that's going to be an issue down the road for China, and I think they're finally realizing some of the negative impacts of the policies that they put in place. The X chromosome carries around 2,000 genes. It's a pretty large chromosome compared to all the chromosomes. But most of the genes on the X chromosome, even though it is a sex chromosome, deal with completely unrelated traits that are unrelated to sex determination. As you can see here, we have like the Duchenne muscular dystrophy gene. So if that's mutated, a child would have Duchenne muscular dystrophy, or ALD, which adrenoleukodystrophy, which was a disease popularized in the film Lorenzo's Oil, if you've ever seen that one. But that's also caused by a mutation on the X chromosome. Neither of those things have anything to do with sex at all, but they're still found on the X chromosome. The Y chromosome, on the other hand, is much smaller. There's only about 78 genes identified on the Y. But the one gene that's super important is called the SRY gene, and you can see the SRY gene located right here on the Y chromosome. Now that determines whether or not you're going to be a male or not. So if you have the SRY present, you will be male. If you do not have the SRY present, you will be female. So in utero, a baby is born, or I shouldn't say that, a baby develops, an embryo develops undifferentiated for the first several weeks of its life. It has undifferentiated gonads. At some point, the SRY is triggered. And if the SRY is triggered, either testes will form, or if it's not triggered, ovaries will form. And after that point, as you can see in this picture or diagram right here, the SRY will trigger the testes to then produce testosterone. And testosterone will then cause all of those other male characteristics, the external genitalia that males have, some of the different bone structure and things like that that males have. All of that will happen because of the testosterone produced by the testes. But the SRY initially caused the testes to differentiate, to turn into testicles. If that didn't happen, if you have no SRY, as in the case of females, then the default would be ovaries forming. And as a result, you would have none of those male characteristics and you would develop into a female. Now, some of the crazy things that can sometimes happen. So the X and the Y chromosome are actually homologous chromosomes, even though they're completely different sizes. And there is some crossing over and things between the two of them. What sometimes can happen is the SRY gene can mistakenly, and it's not supposed to, get crossed over from the Y chromosome to an X chromosome. So you could get a gamete that has an X but has an SRY on it, and a female would seem like they're XX, right? Or genetically they'd be female, XX. But because they have the SRY on the X chromosome, they would end up developing as a male. So a male that was actually XX. There's lots of other things that can happen too in terms of sex determination in humans, like you can have a mutation in your testosterone receptor, and in that case you would have the SRY, you would create testes as a result because you have the SRY. The testes would produce the testosterone, but the cells do not have the ability to recognize testosterone because there's a mutation in the receptor, and if the receptor doesn't properly fit with the testosterone, then it's not going to cause all of the widespread changes inside the cells, and as a result, it will develop as a female. But the problem is you don't have ovaries. You don't have anything to regulate a menstrual cycle or anything like that. So in these individuals, you'd have testes undescended. You'd develop completely as a female your whole entire life until you hit puberty. Then you would not have a menstrual cycle, and you'd wait, and eventually you'd go to the doctor. And they would do a test on you, and they'd say you have testosterone flooding your bloodstream. You have tons of testosterone. So that would tell them that you actually are a male, you're XY. But there was probably some sort of mutation in your testosterone receptor that didn't allow you to recognize testosterone, and as a result, you developed as a female. And at that point, you probably would get, I mean, people have a choice of what they want to do, but a lot of times they'd start to take female hormones and things so that they would develop properly as a female, because that's how they've identified basically their whole life. So there's some information on human sex determination. Now you can also have, as I said, diseases on these chromosomes. And so if you have an X-linked recessive inheritance pattern, as opposed to when we talked about autosomal dominant and autosomal recessive, it's a little bit different the way things are inherited when you have an X-linked disease or an X-linked trait. So you can see here, you can have a mother who's a carrier. Now that mother would not express the disease because she's a carrier, and this is a recessive trait. So she has one functional copy, and the other one isn't functional, but because it's a recessive trait that we're worried about here, the dominant trait will express, and the mother will not express any abnormalities. But the father cannot be a carrier. It's not possible. Either you have the X that has the disease, or you do not, because the Y would not carry that disease. So what happens in this type of inheritance pattern is you can see a male that is unaffected having children with a female that is a carrier. All the daughters would be normal. You could have a normal daughter and a carrier daughter, but the carrier daughter will not have the disease or the trait. But half of the sons will have the disease. So that's one way you can tell it's a sex-linked trait, is if you mate two people that don't have the disease together and only sons are affected and daughters are not, then it's probably a sex-linked or an X-linked recessive inheritance pattern for that particular disease. So the way you can do Punnett scores lots of different ways, and you can use different symbolism to do sex-linked traits. I think the easiest way is to maintain the capital A and the little a and just put those letters on the X or on the X's. So for example, if a mother is homozygous dominant, she's X big A, whoops, she's X big A, X big A, okay? A father is called being hemizygous because he only has one copy of the X, so he can't be homozygous or heterozygous. So he would be hemizygous dominant here, and if you see here, he's hemizygous recessive. This is showing a mother that's homozygous recessive because she's little a on the. Notice we designate the X chromosomes with the little a's. And then a mother that's heterozygous would look like that. She'd be a carrier. Okay? Remember, a father can't be heterozygous because he only has one copy of the X. So examples of these sex-linked traits, one of the most popular ones that's very common in human populations in general, is color blindness. Now, I think most of you, because you are female and you're not as likely to have color blindness, as we kind of talked about before, because mostly the males have it. The only way you can have color blindness if you're a female is your father has to have it and your mother has to be a carrier. So you can see how rare that probably is. So you can see here, I think most of you can see the number 29 here. If you were colorblind, you would just see gray. You would see no difference at all between these different, I don't know why this isn't erasing. There we go. You would see no difference or contrast between these, the dots in that diagram. And that's an X-linked trait. Hemophilia is also an example of this. It was common in some European royal families, specifically the Russian Tsar, the Romanovs. And that is a clotting disorder, and the blood clotting happens on the X chromosome. That's where that protein, or the gene for that protein, is located. And it says 1 in 7,000 males has the allele for hemophilia A, but that doesn't mean they actually have the disease. So it's an even more rare disease than that. And what happens with those individuals is, you can see here, blood does not clot properly. It doesn't form proper bruises. And so even a small bump can actually result in death or a serious consequence. So when I was working at a summer camp, we had a child that was a hemophiliac, and every time he got a bump or a bruise, we'd have to circle it with Sharpie, and if it extended beyond the circle, he would have to have a nurse come in and give him clotting factors so his blood clotted properly. And he always wore a helmet and knee pads, just because those are common places to get bruises and things. So you can see how that definitely has an effect on your life. If you cut yourself, you would just continue to bleed until you got those proper clotting factors. So that is also, hemophilia is another example of an X-linked disorder. So who actually discovered this stuff, which is kind of neat, is Thomas Hunt Morgan. Not a guy that you really have to know the name of, but I'm just giving a little bit of credit for discovering this. And he actually studied sex linkage in fruit flies. And another name for fruit flies, the proper name is Drosophilum melanogaster. And it has 4 pairs of chromosomes, but it determines its sex based on an XY system. Now, he didn't know that at the time. I'm just telling you that now. When we're talking about fruit flies, and you'll see this when we start doing labs and things on this in class, is we use this term wild type to determine the trait that the average fly would kind of have. Usually wild type traits are also dominant traits, but that's not always the case. So in this case, wild type is red eyes. You can see the red eye up here. That's a normal eye color. It also happens to be dominant. There are all these different color eyes that they can have, but red is the dominant wild type eye. And we're going to look at things like white eyes. And white eyes is a recessive trait. So if you look at the Drosophila here, you can see the male versus the female. The female has a pair of sex chromosomes. They're not as differentiated as human ones. But you can see here's the wild type red eye individual, and here's a mutant with white eye. This happens to be a dominant trait, red, as I said, and the mutant white trait is recessive. So let's look at his experiment and how he figured this out. So he took a male with white eyes, as you can see right up here, Now notice there's the Y chromosome. The Y chromosome does not have the I gene on it, and a female that has red eyes. And in doing this, and the female was a true breeding female. These are both true breeding males and females, just like Mendel worked with. In the F1, we ended up getting males and females that all had red eyes. Now this makes sense, right? Because the female has only, for all the males, the female, the female gives a red allele, and the male gives a white allele to the females, okay, to get this situation. And for it to get the male has to give the Y chromosome only, and the female gives a red version of the X, and as a result you get a male that is red eyes. So in this case, all of them have red eyes. So the way we would draw this is something like this. So we'll put let's do the female on top. So she's X big A, X big A, crossed with a male who is X little a, because he has the white eyes, and Y. And as a result, you get X big A, X little a. Same thing over here. So two female carriers and two males that are phenotypically red-eyed. So in this case, every single individual has red eyes. Then we're going to do the F2 now, taking these females and these males and mating them together. So what you get in this case now, remember, the males were X big A, so we're going to change that. And the females are X big A, X little a. And we do the cross. Notice both females are phenotypically normal, although this one's a carrier. A phenotypically normal male, and a male that has white eyes. So you still get a 3-to-1 ratio. If you don't look at sex at all, you get a 3-to-1 ratio of individuals that have it versus individuals that have red eyes versus individuals that have white, a 3-to-1 ratio. But all of those individuals that don't, or that have white eyes, are males. As I said, that's how you can tell if it's a sex-linked trait. Okay. So, if you ever see data on the AP exam, and it shows you in the F2 generation, males having the trait in question, in this case it was white eyes, and females do not, there's still a 3-to-1 ratio overall, but only the males have the recessive trait, then it is always a sex-linked or an X-linked recessive inheritance pattern. So just keep that in mind. That means that the gene is found on the X chromosome. Calico cats actually are another example of this, of sex determination and sex linkage. So only females can be calico, and calico is a specific color pattern in cats. You're orange and black, as well as white too, but white's kind of a separate gene that exists. So orange and black cats as a mix would be a calico cat. So there's an allele for orange fur and an allele for black fur, and there's, as I said, a separate gene that codes for white fur. So males can only be orange or black because they only have one copy of the X, where females can be orange and black. They could still be orange or black if they're like X big A, or X little a, right? But in this case, it would be like an example of codominance, right? So if you're X big A and X little a, in this case, we usually wouldn't designate them as being big A, little a, because it's a codominant situation. So let's do it like this, black and orange. So you would have a mixed black and orange cat if you're a female, but a male can only be either black or a male can be orange. They cannot be both. So there are other methods of sex determination aside from just a normal XY chromosome system. For example, in birds, they use a ZW-ZZ system. Now, you do the inheritance in the crosses the exact same way. The only difference is it is the female that determines the sex of their children, not the males. So females, as you can see up here, are ZW, and males are ZZ. So it's the opposite of what you'd see in humans. But you'd still inherit it the same way. So the females would be hemizygous for the Z chromosome. And males would be, could be homozygous or heterozygous, depending, dominant, recessive, or whatever. Okay, so the ZW-ZZ determination system is just kind of like the opposite, where females determine the sex as opposed to the males. There's other things that determine sex throughout the animal kingdom as well. In some cases, environmental factors determine what the sex is. So in painted turtles, they will lay their eggs in a nest, and there's a certain critical period of time after the eggs are laid that the temperature of the nest determines whether the eggs will be male or eggs will be female. If the temperature is higher than the critical temperature, then they will develop as females. If the temperature is below the critical temperature, it'll develop as males. Now, this is not stuff you have to know for the actual AP Bio exam. They would give you a reading scenario possibly on that, and you'd have to determine that it is an environmental factor that causes sex in this case. But it's always good to have some background on some of these different examples of sex determination in nature. Here's another one. So bees and wasps use a really unique haplodiploid system. And in the case of bees, most of the female of the colony is female workers, and they are diploid. Okay, and any males that are born are just haploid. They're called haploid drones, and a lot of times all they do is mate with females. That's all they do. So they're basically walking sperm, is basically what a male is, and they'll do mitosis to make copies of, or to make sperm when they actually reproduce with the female. So one female actually gets chosen by the hive to be the queen, and she is fed lots and lots of food. It's called royal jelly. So if they're fed enough, they will actually become fertile, where all the other females will not be fertile. The workers are not fertile because they're not fed this royal jelly. And then she will make haploid eggs through meiosis and choose a male reproductive sperm to fertilize her eggs, and then she will make more female worker bees out of that. All unfertilized eggs that she has will develop into males, and the fertilized eggs will develop into females, and eventually they're going to pick one of those females to become her replacement and feed her royal jelly, which is kind of wild. And sometimes they'll even go to another, they'll create a new hive as a result of that process. Even more bizarre is clownfish. Now, Finding Nemo lied to you, because what should have happened after Marlin's wife died is he should have become a female, and then he would have mated with other clownfish, because only female in clownfish is the most dominant female in the chain, or the most dominant individual in the chain is the female. She will lay the eggs. If she dies, the next most largest male, the most dominant male, will become female and then mate with the other males below it. So it's this bizarre thing where they actually change sexes depending on if the more dominant female disappears or dies. Okay, so that's sex determination that we talked about there. There's another inheritance pattern that they show on the exam, and that's when genes are found on the same chromosome. Because if they're on the same chromosome, they actually cannot segregate independently during meiosis. Remember, like if homologous chromosomes line up, they can line up like this or like this, and as a result, if genes are on separate chromosomes, like that, they will line up randomly like that and usually be segregated in a very random pattern. You have a 50-50 shot of getting one or the other. If a gene is linked, though, that's not the case, because the genes are both on the same chromosome. And if they're on the same chromosome, they're more likely to be inherited together. Okay? So for this type of inheritance pattern, the way we test it, our cross is the same. We do a cross where you have two traits here. Okay? Big A, big B, crossed with a little a, little b, little b. So dominant, crossed with the recessive, and as the result of the F1, as always, you'd get a big A, big B, little b. Now that's not anything new. But if we want to test to see if A and B are linked genes on the same chromosome, we would run what's called a test cross, and we would cross the big A, big B, little b individuals from the F1 with an individual that is homozygous recessive at both traits, as you can see here. So this is the classic test cross. You cross it with a homozygous recessive individual, the F1, because we want to see whether these genes are linked or not. And if you do this Punnett square, you'd expect to get these gametes for that big A, big B individual. A big A, big B, a big A, little b, a little a, big B, and a little b. And we've done this before when you watch the Mendelian inheritance video that we made. But if it's linked, you will only get the original combinations of traits that were found in the P generation parents. So you're going to get, as we go back, you're going to get individuals that all look either dominant-dominant or dominant-recessive for the most part. There's only two possible gametes that can come out of it, the big A, big B, or the big A, little b. So if the P generation's genotype was like we said, these are the only gametes you can give. So the A would be linked to the dominant A would be linked to the dominant B, and the recessive A would be linked to the recessive B. So let's do the test cross and what you'd expect to get. Okay, so we're going to do, this individual is going to be the homozygous recessive individual. So the only thing they can give is a little a, little b, right? Because they're little a, little a, little b. Little b, the only gamete you can get out of that is little a, little b. And we said the other gametes are big A, big B, big A, little b, little a, big B, and little a, little b. So what you'd expect to get out of this is a 1-to-1 to 1-to-1 ratio of individuals showing, this is dominant and dominant, right? Dominant, recessive, recessive, and dominant, and then recessive, recessive. A 1-to-1 to 1-to-1 ratio. But what happens in linked genes is we don't get that same ratio. Because remember, the parents were originally big A, big A, big B, big B, little a, little a, little b, little b. So you're only going to get these gametes out of that. And as a result, you should get a 1-to-0 to 0-to-1 ratio. That's what you expect. You wouldn't get any individuals here. And if you see something like that, you know that these genes have to be linked. Now, there's another little twist to this that I'm going to show you now. So this is actually showing you another cross using fruit flies. In this case, they're using fruit fly lingo here. This is just saying like big A, big A, big B, big A, big A, big B, big B, little a, little a, little b, little b. It's the same thing. Okay, they're just using a little bit different symbols to designate it. You create the heterozygous individual. We cross it with do the test cross, okay, with the double mutant as it's called, the recessive. And what you expect to get is a 1-to-1 to 1-to-1 ratio if they were unlinked. But notice what happened. Instead of getting a 1-to-1 to 1-to-1, we got way more of the original parental phenotypes and a few of these other ones. Now I said it should be a 1-to-0 to 0-to-1 or a 1-to-1 to 0-to-0 ratio. So where did these ones come from? Right here. Where did these other individuals come from? Now we have way more of the parental phenotypes, but we have a few of what are called recombinants. And those result from the process of crossing over. Okay? So because the genes are located on the same chromosome, big A, big B, and then I'm going to do this as if they're not replicated. So this is showing the two different homologous chromosomes of a heterozygous individual. Now normally, because they're on the same chromosome, they would segregate together, but if you get crossing over occurring, we get the big B on here and the little b on here. Notice how we crossed over and we actually created a recombinant individual. So the way you can tell a linked gene is way more of the original parental phenotypes and only a few of what are called the recombinants. The number of recombinants, the higher the number, the further apart those genes are on the chromosome. Okay? So that's what this says here. Note, the more recombinants, the further apart the genes are on the same chromosome. And we can actually use that crossing over frequency to determine how far apart they are in reference to other genes on that chromosome. We use this thing called map units, and map units aren't actually a true distance. The distance itself is actually just a determination of crossing over frequency, and the way you do that is by doing this math right here. Map units equals the number of recombinants times 100 over the total number of offspring that exists. So let's use the Morgan example to try to determine the approximate distance apart that these genes are. Okay, so we add all of these offspring together, right? Or here, I should say. They should be the same number, and we get 1,800. The total number of recombinants, 206 plus 185, which is found right down here. We add them together, we get 391. You take 391 divided by the total times 100, and that gives you 21.7 map units, or centimorgans, which is, remember, Morgan's the one that determined this. So he did sex determination, and he also did linkage. So these linked genes are 21.7 map units, which, remember, is just a unit that measures their crossing over frequency. If we increase the number of recombinants that exist, you'd have more map units, and they'd be further apart. If we decrease the number of recombinants, they'd be closer together and less likely to switch and segregate during the crossing over process of meiosis during prophase 1. Sometimes they like to give you chromosome maps, and they'll actually give you like the frequency of crossover over here. So this is basically a measurement of the centimorgans. So it would be 30% or 30 map units or centimorgans. It means the same thing. And they want you to make up a chromosome map from this. So what they're basically telling you, if you look at this, is B and C are the furthest apart at 45% crossing over frequency. And A and D are the closest because they are only 25%. So A and D have to be relatively close to each other. B and C have to be the furthest away. And you can actually make a map doing this. So you can see here, let's check this answer and see if it flies with what we see here. So A and D are 25%, so we're going to write 25 here, okay, between A and D. A and C are 30%. Let's see, B and D, I'm going to go up to these words real quick, are 40. And B and C overall are 45. Now let's see if this makes sense based on, so what's the distance between B and C now that we have this on here? I know it's kind of messed up here, but the difference between B and C is 45, as we can see here. What's the difference between C and D then? Well, this is 30 and this is 25, right? So A and D is 25 and A and C is 30. That means the distance between C and D must be 5. And what's the difference between B and A? The difference between B and A must be the difference of, so if we take A and D is 25 and B and D is 40, then the difference between these two must be 15. And you can actually figure that out that way. So it's kind of just like a little puzzle. Some other chromosomal abnormalities that exist, you can have chunks of chromosomes, change, move, deleted. These are in contrast with, these mutations are in contrast with those tiny mutations we talked about before, which could be very serious, obviously, but like point mutations where you're only changing a few nucleotides. I'm talking chunks of chromosomes here. And we're going to talk about those different mutations in a second. You can also have things called non-disjunction events, which are when the chromosomes don't properly separate during the process of meiosis. That's called aneuploidy also, the resulting having an extra chromosome or one fewer chromosome in a gamete. And then full-scale polyploidy, which is where you have complete non-disjunction events and meiotic errors where you have sperm or egg that are diploid as opposed to haploid, and they fuse together and create 2N, 3N, 4N individuals. Now in humans that's obviously fatal, but in other organisms it can sometimes work out. So let's talk real quick about some large-scale chromosome mutations. You can have a duplication or an insertion. Now it sounds similar to something that we talked about before, having like an insertion or a deletion or something like that. But it's not the same thing. We're talking about huge chunks of DNA. So in a duplication, a whole part of the chromosome is duplicated. Now this can be a problem if too much protein production leads to an imbalance of homeostasis. But if it's not a problem to have these extra genes and maybe producing a little bit of that extra protein, what's neat about it is these chromosomes or these genes are now free to evolve because we actually have an extra copy of these. It's not going to cause any issues. And this can result in the acquisition of new traits. Sometimes those traits are beneficial, and those new traits will actually be selected for. So you can see how this can kind of lead into the process of natural selection and evolution as well. Translocation events, that's when you move one part of a chromosome to another part. They're sometimes called jumping genes, and actually a scientist by the name of Barbara McClintock, she discovered these in corn and won the Nobel Prize because of it. Again, translocation events, you can either have it where you copy it and then move that gene to another chromosome, a whole set of those genes, or sometimes you can just cut it out completely and move it somewhere else as well. And this happens in the body, it just happens randomly. Inversions, that's when the chromosome segment actually flips over completely, and that can happen during the process of crossing over when synapsis occurs and a chiasm is formed. This may not be an issue as long as the breakage is not within a gene. So just flip the genes over. It's still going to read in the same order because we're going 3' to 5', 5' to 3'. There's also deletions where a huge chunk of the chromosome actually is deleted. This is usually happens during the process of DNA replication. And it's almost always deadly. If you're losing several genes, obviously there's going to be a major impact in that. So this is showing you kind of examples of what each process looked like. Here's the duplication event. You can see how we get an extra copy of those genes right there. An inversion right here where they're actually just flipped over. A deletion where you lose part of it. Remember, that's almost always deadly. An insertion where you take part of one chromosome and insert it and create like a larger chromosome out of it. That, again, would only be an issue if that insertion happens between a gene that's important and it disrupts that gene. And a translocation is when you move parts of chromosomes and flip them, like in the case of this. Okay, and this is showing you the process, how some of these processes can sometimes happen through the process of meiosis. Non-disjunction occurs when, as I said, one or more pairs of chromosomes fail to separate during either meiosis 1 and meiosis 2, and depending on when it happens, you can get differences in the gametes that are formed. So if it happens, this is showing normal meiotic division right here. Now remember, in normal meiotic division, you want to have all sperm and egg that are N from the 2N individual that started at the beginning. But you can see if meiosis, if non-disjunction happens here, where the chromosomes aren't separated, you're going to create 2 gametes that are N+1 and then 2 gametes that are N-1, meaning this has an extra chromosome here and this has one less chromosome there. And in the case of having non-disjunction in meiosis 2, you're going to have this normal one over here, which creates N sperm and egg. But over here, you're going to get the N+1 and the N-1. So obviously, having it happen during meiosis 1 is more severe and not as often. It's not as severe in meiosis 2 because you still get 2 normal gametes out of it. But if this happens, these happen to be the sperm or eggs that cause the problem, then it's not going to matter either way. So one example of a non-disjunction event is Down syndrome, and that happens when there's non-disjunction in the 21st chromosome, and that results in a gamete that has either 2 copies of the 21st, a gamete has 2 copies of the 21st chromosome when it should only have 1, or an extra piece of that chromosome happened because of a translocation event or something like that. And then when the diploid gamete fuses with a normal haploid gamete, it results in 3 chromosome 21s, hence the trisomy 321. And as a result, you get, of that excessive protein, you get the characteristics of Down syndrome that are common, that we commonly see. So examples of that would be like low muscle tone, sometimes issues with the tongue attachment, the separation of their eyes, things like that, and lowered IQ and stuff. So this actually shows you right here how you have a sperm and an egg fusing together that have too many chromosomes, and as a result, the 21st chromosome would be this blue one right here. You have 3 copies of it, and as a result, you'd have Down syndrome. Edwards syndrome is trisomy 18. Most trisomies result in death. Even Edwards syndrome, miscarriage count is extremely high, and if miscarriage doesn't happen, the chance that they die very early on in their life is extremely high. As it shows here, only 1% survive to age 10. So you can see how these trisomies are very serious issues, having these extra chromosomes and extra proteins. As I said, though, most trisomies of other chromosomes, the 18 and the 21st chromosome tend to be very small, do result in automatic miscarriage. Sex chromosome non-disjunction, though, is a little bit more interesting because if you have extra X's and Y's, it tends to not have as big of an effect. Now, it can have an effect, obviously. So let's talk about 3 examples. So one's Klinefelter syndrome, where you'd have non-disjunction of the X, and you'd be XXY. So you would have 2 X's like a female, but you also have a Y chromosome like a male. And because if you have the SRY gene, the way I remember this, SRY, you're a guy, you will have males that happen to be phenotypically taller than normal, but they'll have some feminized characteristics such as wider hips. They'll have breast development. Those males are also usually sterile. So you can see that there's obviously issues with having this extra X chromosome. In Turner syndrome, you only have 1 X chromosome and nothing else. So you'd be X. That's it. In that case, because you have no Y, you're a female. And most X0s, or Turner syndrome individuals, result in miscarriage. But if they do survive, they tend to be sterile, and they have unfeminized characteristics. They tend to have a little bit more boxy of a shape as opposed to, the standard, wider hips that females do. And then phenotypically normal males would result from an XYY, just having an extra Y chromosome, which is called XYY. Originally, they thought that XYY could cause violent behavior, as there were several serial killers that actually were XYY, but that has since been discredited. Polyploidy is like non-disjunction, except it's a full set of extra chromosomes. In humans, always deadly. In most organisms, always deadly, but plants have a tendency to be able to handle this and actually create new species spontaneously through polyploidy. There are a few animals that can do this, but it's much rarer in animals, specifically vertebrates. And you can see here you actually have, these are diploid individuals, these are triploid individuals, and these are quadruploid individuals. And here's showing you some examples of the mustard family of plants. And you can see how through these extra chromosome events, through this polyploidy events, you end up getting these different species that result in, because of these having extra chromosomes and things like that. So now we're going to look real quick at some karyotypes, at all of these different things, to figure out what information can we get from a karyotype to determine sex or to determine different diseases. Now we know what causes these different diseases. So let's look at this first one right here. So this is showing a human male karyotype. And we know, because we see 2 of every single chromosome, 1 through 22. And then down here we see an X and a Y, a large and a small chromosome. So we know it must be a male. A female has every single pair, 1 through 22, but you'll have 2 X chromosomes down here. So we know it has to be a human female. Now let's look at some diseases. We're going to look at all the pairs here, 1, 2, 3, 4, 5. Everything looks good until we get to the sex chromosomes. Notice there's a Y and 2 X's. So this is XXY. This is an example of Klinefelter syndrome, which is XXY. Remember, phenotypically taller than normal males, but with feminized characteristics. How about in this one? Again, all the sets are there, except we only have 1 X chromosome and no Y. So this is X0. This is Turner's syndrome, which causes females that are phenotypically shorter than normal and are usually sterile. If we look here, we look throughout all the chromosomes. We check the sex chromosomes. We have 2 X's, so this is a female. There's no extra Y, so we're good there. But if you look over here, we have 3 copies of the 21st chromosome, and thus this is someone that has Down syndrome. And they can do this. They can actually do pre-screening. They can do an amniocentesis to determine whether or not the baby would be trisomy 21 or trisomy 18, and to help prepare families for to help someone that has Down syndrome and help develop them into adulthood. the psychological parts of having to deal with that, because people who have Down syndrome tend to die much younger than everyone else, because they tend to also have heart conditions. They usually have a hole in their heart when they're born that has to be surgically repaired. They usually only live into their 30s. People can live into their 60s and 70s sometimes, but it's much more rare because of the heart conditions that they have. So this last one you can see here, trisomy 18. This would be an example of Edwards syndrome. All right, so that is chromosomal inheritance. If you have any questions, let me know.